6. Some of the cells in the pancreas secrete proteins. The diagram represents a pancreatic cell. The structure labelled Y represents the... Okay, so first things first, they've said a pancreatic cell, but a pancreatic cell means it's in the pancreas, and that's in the human body. So basically they're saying a eukaryotic cell. And you've studied the contents, so the organelles that are present in eukaryotic cells, but the word here, pancreatic, is just to throw you off. They could have replaced it with any other cell in the human body and given you the same contents that are found everywhere in eukaryotic cells. So just ignore the word pancreatic cell. They're just asking you what are the contents in a cell. So, structure labelled Y. That's this. Okay. So you would have seen this image in a textbook, but maybe not in this type of outside shape, because that's supposed to be representing a pancreatic cell, but the internal structures you would have come across. And so let's start to pick them out. So these structures, especially when you've got these two quite close together, this represents the smooth endoplasmic reticulum, and this is the rough endoplasmic reticulum with ribosomes attached. And typically it's got this structure next to it, and this distinct shape with the vesicles around it indicates that this is the Golgi body or the Golgi apparatus. And these rod-shaped structures are mitochondria, meaning that what's left over is X, and X is just a cell membrane. And so Y would be the rough endoplasmic reticulum. The structure also found in a prokaryotic cell is labelled. Okay, so we know that structures like smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi apparatus, these structures are always found in eukaryotic cells, and so automatically we can get rid of them. And mitochondria also in eukaryotic cells, meaning the only thing that's left over for us is X, which is the cell membrane, which is found in prokaryotic cells as well, meaning the answer is Y. Oh, sorry, the answer is X, XOB, yeah. Okay, calculate the magnification of the cell. Okay, so when it comes to magnification, there's a little thing I like to remember, and it's this triangle. And I always try to remember I am. Yeah, I am. And the I stands for the image size. And the A stands for actual size. And the M stands for magnification. Okay, and so you've come across triangles before in mostly physics where you cover up the thing that you want. So here the magnification, we cover it up, and so we know that in order to get magnification we have to do the image size divided by the actual size. And the image size is the one that you measure by a ruler, and the actual size is the one that's always given to you. So here they've given us 10 micrometers. The length of the actual pancreatic cell is 10 micrometers, and so automatically that's going to be our division, I over A, and A we have as 10 micrometers. Now we need to standardize the measurements. So image size, it's always you getting a ruler and measuring this by a ruler. I always recommend having a ruler with millimeters on it, because you'll never have a ruler with micrometers. You'll have a ruler that's centimeters or millimeters, and having a millimeter one is easier because you can convert millimeters to micrometers by times by 1,000. And times by 1,000. Yeah, you times by 1,000. And so I can't actually measure it here, and the thing is, because when you have this in exam, you'll have a standard paper form printed out, but because I have copied and pasted this, I can, if I make this size, then my measurement with a ruler is going to become quite small. And if I increase this size, then my measurement with a ruler is going to appear bigger, and so it's not quite standard. And so we'll just imagine that I've measured this with a ruler, and say I got something millimeters. I will then times by 1,000 to convert that number into micrometers, and then I will divide that number by 10 to get the magnification. But the thing is, you need to remember is this: I am. The actual size they will always give to you in a calculation if they want you to calculate the magnification. All then you have to do is get the image size by measuring it with a ruler, and that's it. Sometimes they can ask for the actual size by giving you the magnification and you having to measure for the image size, and that would be it. Now, explain why these pancreatic cells contain large numbers of organelle Z. Okay, so if we go back, organelle Z, we said this is the mitochondria. So what this question's asking, it's not actually asking you, well, why does the pancreatic cell need organelle Z? Because the chances are that you've never studied the pancreatic cell, and actually this question is basically asking you, what is the role of organelle Z? That's it. So it's about having the skill to see, well, what does this question actually want? Because it's always something you've studied. So actually this question is just asking you, why do you need organelle Z? What's the role of organelle Z? And you've studied the role of mitochondria. And the first point is, well, mitochondria produce ATP. It's the main thing. It produces ATP. And well, why is this important? Remember, when it's an explain question, it's always why. So why is this important? Well, because this will lead to organelle Z supplying energy for the synthesis of protein. So for protein synthesis. So supplies energy for protein synthesis. And hence it makes sense why mitochondria is found next to the rough and the smooth endoplasmic reticulum, because the ribosomes here will also undergo protein synthesis. And so you have the mitochondria near to supply the energy. And so supplies energy for protein synthesis, and also supplies the energy for the transport or the modification of proteins, and hence why it's also found next to the Golgi apparatus. So also supplies energy for modification and transport of proteins. Moving on. The secreted proteins are made from amino acids. Two amino acids join together to form a dipeptide. Draw a diagram to show the structure of a dipeptide. Okay, so a dipeptide is two amino acids joined together, but what they're testing here is the ability to know how to draw a peptide bond. And so you know what two amino acids look like. So if we draw this, an amino acid is a central carbon atom with an R group, hydrogen, and here I'm going to do the amino group, so the NH2. Notice how I've written it like this, NH2, because it's actually the carbon that's connected to the nitrogen atom. Don't be tempted to do this, because that indicates that the carbon's attached to the hydrogen atom, and that is wrong, and you can sometimes lose marks for that. And so this will be connected to a carboxylic acid group. However, when it's a dipeptide, and that means then one amino acid has joined with another, you have a condensation reaction happening, and so you get removal of water, and that means the carboxylic acid group becomes just a carbonyl group. And so this is what we have. Now, drawing the other carbon atom, we're going to have the joining with a amino group, and we're going to have the removal of a hydrogen atom, because now we have one atom of hydrogen removed here, and oxygen and hydrogen removed here. That means one molecule of water has been removed. And that's for every single reaction that occurs. You always, a condensation reaction that occurs, you always have a removal of water. Now let's draw the remainder of the amino acid. And that will be it. You have shown the groups, you have shown the peptide bond, and you have shown the R groups and the hydrogen groups, and that's it. That's 3 marks. That's a nice easy question here. And that's it.